Hello, everybody, and welcome to a most requested series of classes for the very special class 12th batch special Cbse Term 1 one-shot revision of all the 5 chapters sexual reproduction in flowering plants. In today's class, in a one shot, in next 1 hour, we are going to quickly recap all the important points, all the important diagrams in the NCERT, each and every minute points in the NCERT we are going to discuss in the next 45 to 1 hour. Okay, so just get into the session, and today we'll have the very first unit, reproduction unit, and the very first chapter, that is sexual reproduction in flowering plants. Not only for the Cbse Term 1, the same session will be useful for the NEET 2022 aspirants as well. So let's get into today's class. Those who are watching the session, don't forget to hit the like button, do share the video to your friends, and subscribe the amazing platform for more updates on biology. Today we are having the Cbse Term 1 sexual reproduction in flowering plants on 12/12/2021. And this is your super amazing upcoming schedule. That is, today you'll have the very first unit. 13th, that is tomorrow, you'll have human reproduction. On 14th, you will have the reproductive health. 15th, principles of inheritance, and 16th, molecular basis of inheritance. Okay, let's get into today's topic very soon. Yes, everyone. So in this chapter, we are going to first discuss about what is a flower and how flower help in the process of sexual reproduction. So as we all know, in case of human beings, there is a separate male reproductive system. There is a separate female reproductive system. So exactly as in the case of plants, you will find that flower is the modified stem, which is actually acting as the reproductive organ. Similarly, in case of human beings, in case of plants also, you will find separate male as well as separate female reproductive systems. Yes, everyone. So let us read one by one and let's go to the session. Flower is a modified stem. So flower is a modified stem which functions as the reproductive organ. That is, if the flower is a female flower, then it produces ova, that is the egg. And if the flower is a male flower, it will produces what? A pollen grain. So typically an angiospermic flower, that is from your lower classes onwards, you have been studying the same, right? Flower will be consisting of two main structures of flower. The parts of a flower can be divided into two. I'll write it once again with the pen. Just listen. Yes. So a part of a flower can be mainly divided into two. That is, you can call them as the essential wall, and you will have the non-essential wall as well. What do you mean by essential wall? Essential walls of a flower means that those parts of a flower that is actually helping in the process of reproduction in case of the plants or in case of the flower. So those structures, you will call them as the essential parts of a flower, and they are of two types, or they are consisting of two different parts, that is the androecium in case of males and gynoecium in case of the female flower. Okay, so once again repeating all the important points, flower is a modified stem which functions as a reproductive organ. So if it is a female flower, it will produce as egg, and if it is a male flower, it will produce as pollen grain. There are mainly 4 whorls of floral appendages associated with a flower, that is the calyx, corolla, androecium. What is androecium? Androecium is actually the male reproductive part of the plant, and gynoecium is the female reproductive part of the plant. Coming on to the structure of androecium, you will find that they'll have a bilobed structure like this. They will have a bilobed structure like this, and they will have a slender, thin stalk-like structure as well. So this bilobed structure, you will call them as the anther. What you will call them as? You'll call them as the anther, and the stalk-like structure, you will call them as the filament as well. Clear? So that is the anther and the filament. When coming on to the female reproductive part of a flower, you will find a receptacle-like area where you will find that the pollen grains will be received at that particular point of place. So this is the stigma, which is acting as the receptacle or the recepting area for the collection of pollen grains or for the landing of pollen grain. This long tube-like structure over in the female reproductive part, you will call them as the style. What you will call them? You will call them as the style. And the bulged bottom portion of the female reproductive part of a flower, you will call them as the ovary as well. So these are the main parts of an androecium and a gynoecium. So let's discuss. This is a very first diagram in your NCERT. So be careful that you read each and every line of the NCERT, as well as you pay attention towards each and every labelings on the diagram mentioned in the NCERT. Yes, mainly in coming on to the case of reproduction, you will find there are mainly three events, that is pre-fertilization events, fertilization events, and the post-fertilization events. So in the pre-fertilization event, as you have already studied that, in the pre-fertilization event, you will find actually the events that are happening prior to fertilization. What are those events? It includes the gametic formation or the gamete formation, as well as the transfer of the gamete. If it is like male and female flowers are different, then the male gametes have to be transported into the female gamete, right? So for that, transfer of pollen grains must take place, or transfer of gametes must take place during the pre-fertilization event. So let's discuss. This is a very important line which have been mentioned in the NCERT. That is, several structural and hormonal changes lead to the formation and development of a floral primordium. Inflorescence is formed that bears floral buds and then the flowers. Androecium is the male reproductive part and gynoecium is the female reproductive part, in which the respective type of gametes are formed. So that's an important statement. Coming on to the next topic, that is stamen, microsporangium, and the pollen grain. Here I have to tell you that pollen grain is not actually the male gamete. It is actually the male gametophyte. Okay, that should be kept in mind. It is not the male gamete, it is the male gametophyte. Okay, so stamen consists of, as I've already mentioned, long and slender stalk-like structure that is called the filament, and a bilobed structure which is called the anther. The anther is actually a four-sided structure. So guys, this anther, when you cut it, that diagram is also mentioned in the NCERT, you will get a structure like this, where you are studying what the four wall layers you are studying using this diagram, right? So this is actually the structure. So it consists of a four-sided structure consisting of four microsporangia. Look, four lobules are there in each. Okay. Microsporangia develop further and become the pollen sac, which contains the pollen grain. So inside each of these, this must be filled with millions and millions of pollen grains. Okay, so this microsporangium, so this is a microsporangium. This microsporangium will enlarges and it will matures to form the pollen sac, inside which pollen formation takes place. Clear? So this is the brief idea about the stamen, Microsporangium and the pollen grain. Microsporangium is generally surrounded by 4 wall layers. Okay, Microsporangium is generally surrounded by 4 wall layers. That is, look at this diagram. You'll have the outer epidermis layer, which is followed by endothecium. Then you will have the middle layer. Then you will have the microspore mother cell. That is, there are mainly 4 layers. So I'm marking only the 4 layers. That is, the 4th or the innermost layer is the tapetum. Clear? So epidermis from outer to inside. If they are asking, first one is the epidermis, second one is the endothecium, third one is the middle layer, and fourth one is the tapetum. That's very important. The first 3 layers, that is the epidermis, the endothecium, and the middle layer, actually help in the process of dehiscence of anther and release of pollen grain that have been mentioned in the NCERT, so not specifying it again. Whereas the function of tapetum is very important. Tapetum nourishes the developing pollen grain. That is, tapetum is actually nutritive in function. Tapetum is actually nutritive in function, whereas the first 3 layers, that is epidermis, endothecium, and middle layers, all the 3 layers will help in the protection and dehiscence of anther to release pollen grains. Clear? So these are the 4 main wall layers. That was an easy topic, I hope. Next, inner to this tapetum, you will find sporogenous tissues. Okay? It is a compactly arranged homogeneous cells which are present at the center of each microsporangium when the anther is young. So if the anther is young, that is in case of the younger stage of the anther, that is in case of the young anther, you will find inner to the tapetum, you will find what? The sporogenous tissue. What you will find? You will find the sporogenous tissue as well in them. Clear? So the question may be, what are the 4 layers? That is the epidermis, the endothecium, middle layer, and the tapetum. And if they're asking you what is if they're asking you 5 labelings and you're asked to write from outside to inside, including sporogenous tissue is also given over there, then you have to write that sporogenous tissue is present towards the inner of the tapetum. Clear? Yes. Next, from the beginning onwards, we have been telling, like in case of plants, just listen. In case of plants, you will call the male gametes as microspore. What you will call? You will call them as the microspore. And you will call the female gamete as the megaspore. What you will call them? You'll call them as the megaspore. So the process of formation of microspore, you will call them as microsporogenesis, and the process of formation of megaspore, you will call them as megasporogenesis. So since we are learning about the male gametogenesis, you'll study about the process of microsporogenesis. So what is actually microsporogenesis? Microsporogenesis is actually the process of formation and differentiation of microspore. What is actually microspore? It is a pollen grains from microspore mother cell by reductional division. What is reduction division? Reductional division, as you have studied in the chapter cell cycle and cell division, you're thoroughly aware about that. It is what? Meiosis. So the reduction division is meiosis. So inside your NCERT, you will find the statement like this: the process of formation of microspore, that is pollen grains from microspore mother cell or MMC or pollen mother cell or PMC by meiotic division is called microsporogenesis. So what is the type of cell division which is seen in case of microsporogenesis? It is the meiotic division or the process of reduction division. The cells of sporogenous tissue undergo meiotic division to form a microspore tetrad. As the anther mature and dehydrate, the microspore dissociate and releases the pollen grain. Is this clear? I will show you, I'll give you a clear idea about that. If yes, just listen, I'll give you a clear idea about that. That is, this is the anther. First of all, you're getting an anther. What happens is that the anther will divide into 2. Okay, the anther will divide into two. After some time, that is on maturity, this anther will again start dividing and it will convert themselves into four. Oh my God. Okay, this will again divide into four like this. And each of this layer will develop its own wall layer. Okay. Each of this layer will develop its own wall layer like this. Okay. Wall layer like this. And what is this condition? You will call them, this condition you will call them as the microspore tetrad. That is, four different microspores are present. Microspore tetrad. Clear? Microspore tetrad condition. And what happens finally leads to the formation of four pollen grain. It leads to the formation of four pollen grain. So for the CBSE board exam, you can expect one numerical type question from here. Even though this is the biology part, you can expect a numerical problem from this chapter. Okay, like how many pollen grains will be formed by 100 mitotic division, 100 meiotic division and all, they'll be asking you. Okay, so during one microsporogenesis, you will get four pollen grain. You will get four pollen grain. So one mitotic division, four pollen grain. So suppose if the question is 100 meiotic division are going to happen, how many pollen grains are formed? 400 pollen grain. That is by one, you'll get four. So by 100, you'll get 400 meiotic division. Is this clear? Is this process of microsporogenesis clear for everyone? Yes. Next, we are moving on to the process of or into the detailed study of pollen grain. So guys, when you're coming on to the structure of a pollen grain, you'll find that pollen grain represents the male gametophyte. Pollen grain represents the male gametophyte, and the pollen grains are made up of two layered wall. That is, suppose if this is the pollen grain, you'll find that their outermost structure will be an irregularly shaped structure, which you will call them as the exane, and this exane layer is made up of a very complex and a very important material. The name is very important, sporopollenin, by which you are preserving the pollen grain. Sporopollenin, made up of sporopollenin, which is actually the most resistant organic matter known. It can withstand high temperature, strong acid and alkali, and there is no enzyme that can degrade the sporopollenin. That is the main reason why you are preserving, or why you can preserve the pollen grains for so long. Whereas the inner layer, that is the intine, is a very thin and a continuous layer. Very thin and a continuous layer. Whereas exane is irregular and non-continuous. That is, in between you'll find small spaces, which you will call them as the germ pore. So sometimes a question can come. Germ pores happens on the surface of intine. No, it happens as a part of exane. Irregularity in the exane layer causes what? Germ pores made up of cellulose and pectin. So the intine layer is made up of cellulopectin, whereas exane layer is made up of sporopollenin. Next is the germ pore. That is, as I've already mentioned, germ pores means that those area on the exane where sporopollenin is absent. Very important. So those parts of the exane where the sporopollenin is absent, you'll call them as the germ pores. And what is the function of this germ pore, or what is the importance of this occurrence of the germ pore? What is its function? It is actually helping to form the pollen tube during the process of pollen growth. Clear? During the process of pollen growth, you'll find the formation of pollen tube through the germ pores. And as in all cells, you will find the plasma membrane is actually surrounding the cytoplasm of the pollen grain as well. Clear? Okay, everyone. Okay, everyone. So let's move with the next one. Yes. As we have already done, a mature pollen grain consists of two cells with a nucleus. I have already explained that for you. That is a pollen grain. The outer side will be the exine, inner layer will be the intine, and inside the intine, you will find two different cells. What are the two different cells in case of a mature pollen grain? You will find a vegetative cell, you'll find a generative cell. So the larger cell you will call them as the vegetative cell, and the smaller cell you will call them as what? The generative cell. So vegetative cell is the bigger cell. It is consisting of the abundant reserve food material. It consists of the large nucleus, a large irregular nucleus. Very important term, large irregular nucleus. It is a bigger cell inside the pollen grain, responsible for the development of the pollen tube or the development of the pollen grain. Whereas generative cell is a small that is involved in the formation of male gametes. So they can ask you a question, which of the following cells, generative or vegetative cell, helps in the process of formation of the pollen tube? Pollen tube is actually formed by the contents of the vegetative cell. So pollen tube is actually the result of vegetative cell, whereas which cell help in the process of fertilization, that is the male gametes are formed. Male gametes are formed by the mitotic division happening in the generative cell. In the generative cell, what is its speciality? It consists of dense cytoplasm and nucleus. It consists of dense cytoplasm and the nucleus. Yes, this is the last small paragraph that have been mentioned in the NCERT as well. Pollen grains of many species, especially carrot grass or parthenium, causes several allergy and bronchial diseases in some people. Sometimes you can use these pollen grains as nutrients or as pollen tablets for food supplements. And this viability, there are pollen grains that are viable for months. There are pollen grains that are viable for only 2-3 days. There are pollen grains that are viable for more than a year as well. What is the speciality of the pollen grain that makes us viable for this much long time? It is actually what? It is the sporopollen, because nothing can degrade the particular material, right? Yes. And name the process by which you can preserve this pollen grain. Name the process by which you can preserve this pollen grain. It is cryopreservation. What is it method? You will call them as what? Cryopreservation. Cryopreservation means you're going to store the pollen grain in liquid nitrogen at a temperature. Okay, it's minus 196. Minus 196 degrees Celsius in liquid nitrogen. Clear? So that method you will call them as cryopreservation. So that's all about the male gamete and all points regarding the pollen grain, microspore, microsporogenesis, anther, and filament. That is, androecium is over. Now we are moving on to the detailed study of the part which you will call them as the gynoecium. First, we have to study about the pistil, megasporangium, and the embryo sac. And next, we have to study about the process of megasporogenesis and the structure as well. Clear? So let's discuss about the pistil first of all. So gynoecium may consist of a single pistil. Gynoecium may consist of a single pistil. What do you call the single pistil? Monocarpellary. Or it may consist of more than one pistil. What is it? It is polycarpellary. Sometimes if the pistils are fused, you will call them as a syncarpous condition. If the pistils are free, you will call them as the apocarpous condition. Very important. Monocarpellary, single pistil, more than one pistil, polycarpellary. If they are fused, it is syncarpus. If they are free, it is apocarpus. This was a NEET 2021 question. Okay, NEET 2021 question where the students have not paid attention towards this part of the NCERT because this is not mentioned in the NCERT. It's just given there as a diagram. Just as the objective or as the detailed explanation of the diagram, they have just given you as a subheading over there, but nobody just pays attention towards the subheadings. What does diagram indicate over there? Okay, so multicarpellary syncarpus. These statements are very important, so pay attention, guys, everybody. Multicarpellary, oh my god, I think you will not be able to see that. Multicarpellary syncarpus, that is more than one carpel fused pistil, is found in papaver. Where you can find that? You will find that in papaver. And the multicarpellary apocarpus pistil you will find in Michelia. Where you will find that? In Michelia. Clear? And as I've already told you, every pistil consists of stigma, style, and ovary. That is the very first portion where the pollen grains land during the process of pollen pistil interaction. Is actually the stigma, the long portion is the style, and the bulged portion is called the ovary. So stigma, style, and ovary. Inside the ovary, you are going to find what? The ovules as well. Clear? Is this topics clear for everyone about the pistil, about the megasporangium, and about the embryo sac? Yes, I hope it's done. Let's move with the next one. Megasporangium. So this is a very easy one that you have been common with, Megasporangium. Just I'll be highlighting that all these things. So guys, this is the structure of an embryo sac. The ovule is attached to the placenta exactly as how an embryo, that is how a zygote is getting attached, how a zygote is getting attached to a placenta of the humans. Exactly like that. In case ovule where the development is going to happen, the ovule is going to get attached with the placenta, like the small structure is attached with the placenta itself. The stalk by which it is getting attached to the placenta, you will call them as a funicle. So funicle is the stalk by which the ovule is attached to the placenta. The part of the ovule where the funicle attaches with the ovule, you will call them as the hilum. So hilum is the region where the funicle gets attached with the ovule. The protective covering around, this is a protective covering. So these are the protective covering. So these protective covering, you will call them as the integuments. The area or the part of the ovule where the integuments are absent, you will call them as the micropylar end. You'll find them as the micropylar end, and the opposite portion to the micropylar end, you will call them as the chalacel end, which is actually the basal part of the ovule. Just when we study about the structure of a microspore, there you studied about tapetum. What was tapetum? It was the nursing cell over there or the nutritive cell over there. So exactly in case of microspore and megaspore as well, in case of megaspore, you will find another nutritive tissue. In case of female, that is the nucellus tissue. And what is the ploidy of the nucellus, guys? From this chapter, you can expect a question from the ploidy part. Ploidy of endosperm or microspore mother cell or primary spermatocytes from the next chapters. So either from botany or zoology, you can expect what? Ploidy type question as well. A direct MCQ from these terms also you can expect. Maybe it may be exactly chalasa or the name of the cells as well can be asked for your exam. So please be careful while you discuss with this chapter or while you study with this chapter. Clear, everyone? Let's continue with this. Okay, let's continue. Megasporogenesis. What is actually megasporogenesis? The process of formation of megaspore from megaspore mother cell by the process of meiotic division is known as megasporogenesis. So what is megasporogenesis? The process of formation of the megaspore from the megaspore mother cells. That's very important. From where? That is from the megaspore mother cell. By the process of meiotic division, which takes place inside the ovule, you will call them as the process of megasporogenesis. The ovule differentiates into a single megaspore mother cell in the micropylar region. It will undergo meiotic division that results in the production of 4 megaspore. But in case if it was in the case of microsporogenesis, these 4 microspores are going to get it expressed at the end. But in case of this female gametogenesis over here, what you will see is that from the MMC, that is by the megaspore mother cell, 4 megaspore will be formed, but 3 towards the micropylar end. 3 towards or 3 megaspores will disintegrate, where only 1 will be left out. What happened? Only 1 will be left out. And this 1 will undergo sequential mitotic division, and finally it will result into an 8 nucleated 7-celled condition. It will result, this is very important, 7-celled, 8 nucleated condition. What is it? It is 7-celled, 8 nucleated condition. Very important. So we have already discussed the process of microsporogenesis. Here we are studying about the process of megasporogenesis. From the megaspore mother cell, you're going to have, through meiotic division, the formation of megaspores. So as a result, during the divisions, 4 megaspore will be formed, but 3 of the megaspore will degenerate or disintegrate, and only 1 megaspore develop into a female gametophyte, which you will call them as what? The embryo sac. So what is the, let me take out a slide for you. Yes, over here. What is it? Yes, what is it? You'll find that the male gametophyte, I'm going to talk about not about the gamete, but about the gametophytes. Okay. Yes. In case of the males and in case of the female. Very important question, but you will get it confused. So that's the reason why I'm writing it over here. In case of males, the male gametophyte will be what? It is the pollen grain. What is it? It is the pollen grain. And what is it? Gametophyte in case of the female, it is the embryo sac. What is it? It is the embryo sac. So pollen grains are the male gametophyte and embryo sac is actually the female gametophyte, and it's not the gametes. Clear? So that is what I have told you. 3 cells towards the chalacal end, you will call them as the antipodal cells. 3 cells towards a micropylar end together, you will call them as the egg apparatus. That is, the egg apparatus will consist of a central large cell called the egg and 2 surrounding cells called the synergids, and along the center of the embryo sac, you will find a double nucleated structure, half fused structure. Clear? That you will call them as a secondary nucleus or the primary endosperm nucleus. Clear? So this point is very important. 3 cells grouped at the micropylar end to form the egg apparatus, 3 cells at the chalacal end to form the antipodal cells, and at maturity embryo sac is 7-celled, 8 nucleated condition. So this is the exact diagram. You must pay attention. All that is belonging to the antipod, sorry, towards a micropylar region, that is a central large cell is egg along with 2 surrounding synergids. The central cell, 2 polar nuclei half fused, and you will find the antipodal cells as well. Clear? This is a final diagram. Sometimes this can come as a case-based question. Sometimes not sure about the same. Since you have the case study question, this may be asked for the exam. Clear? Is this topic clear for everyone? Yes, everyone? Yes. So that's all about the process of gametogenesis, that is a microsporogenesis and the megasporogenesis. Next is the pollination. What do you mean by pollination? Pollination is a transfer of pollen grain from the anther to the stigma. Transfer of pollen grain from the anther to the stigma. Okay, that can actually happen in various methods. Right. So the very first method which you are going to study is over here is autogamy. What do you mean by autogamy? It is a transfer of pollen grain from the anther to the stigma of the same flower. Very important point. Okay, what is autogamy? Transfer of pollen grain from the anther to the stigma of the same flower. That is from one flower to the same flower itself. That is both male and female will be present in that itself. Suppose this is a flower. Male gametes will fall on the same female flower itself. Okay, just give me hearts on the screen, everyone. Okay, yes. There are mainly two types of flowers. That is a cleistogamous flowers and the chasmogamous flower. Cleistogamous flower means the flower which does not open at all. Example is a viola, oxalis, camelina. Very important three words. Viola, which is a common pansy, oxalis, and the camelina. These flowers will never open at all, and the cleistogamous flower are always autogamous. So I'll erase everything and I will write for you that. Okay, so the flowers which does not open at all, that you will call them as what? The cleistogamous flower. The cleistogamous flower are always autogamous, as there is no chance of pollen landing on the stigma. Very important, another point I have to mark it for you. Just write it down. Cleistogamous flower produces assured seed set even in the absence of pollinator. Very important. Cleistogamous flower produces assured seed setting even in the absence of a pollinator. And the examples also I've mentioned earlier itself, that is a viola, oxalis, and the camelina. Clear? Chasmogamous flower means they have exposed anther and stigma. They have exposed anther. That is, they are open flower. They are what? They are open. Clear? Whereas cleistogamous flower means they are closed. And its example is the viola, oxalis, and camelina. Very important. For your model examination, for many schools, you might have expected this question. So similarly, this can be your board exam question as well. So autogamy is the first method of pollination. Similarly, geitonogamy is there and xenogamy is also there. Autogamy is actually the transfer of pollen grains from the anther to the stigma of the same flower like this, right? Same flower. But in case of geitonogamy, what you will see is that the transfer of pollen grain from anther to the stigma of different flower of the same plant. Suppose, one second. Suppose this is a plant, this is a flower, this is another flower of the same plant. What does this process of geitonogamy means? That the pollen grain from this flower will fall into another flower of the same plant. But what was in case of autogamy? Autogamy means even though so many flowers are present also there, if male gametes are originated in this flower, it will fall onto that flower itself. There is no transfer. And that's the best example for an autogamous flowers is the cleistogamous flower. Which are those flowers? They will never open at all. That is a common pansy, oxalis, viola, and the camelina. And what is xenogamy? Xenogamy means it is a complete transfer of an anther from the male part to the stigma of another flower of another plant. Clear? Maybe of the same species. Okay? Maybe of the same species, but not in the same flower itself or not in the same flower of the same plant as well. Not even in the different flower of the same plant also. It may be in different flower of a different plant. Clear, everyone? And you have studied about three major agents of pollination, that is wind, water, and the insect. So what are the importance of wind pollinated flowers? Their pollen grains will be light and non-sticky. They will have wings, well exposed anther, large feathery stigma to catch up this pollen, one ovule. So sometimes this can be a question. Name the type of pollination seen in a plant which have only one ovule, which is that wind pollination. Okay, and these will be arranged as inflorescence. For example, corn cob and date palm, you will find what? Wind pollination. So wind pollination, what are the characteristics? Dry, non-sticky, light, well exposed, large and feathery stigma, well exposed anther, only one ovule will be present. Whereas coming on to the case of water pollination is mainly seen in bryophytes, pteridophytes, and algae. The pollen grains will be protected. Water pollination, what is the speciality? The pollen grain will be protected by a mucilaginous covering. What is it? Their pollen grain will be covered by a mucilaginous covering. For example, freshwater plants such as Vallisneria and Hydrilla and seagrass, which is a Zostera, they'll also shows what? Water pollination. Their pollen grains will be large in number in order to compensate those pollen grains which are lost as a result of water flow. And this water pollinated flower will never produce nectar or any other sweetie materials. Clear? Clear, everyone? Third one is the insect pollination. Okay, what is insect pollination? Similarly, very first time itself, the point that comes to your mind will be like the flower will be very highly attractive. It will produce nectar. Stigma will be sticky. That is, the pollen grains will be sticky over there. Pollen grains will also be sticky, whereas a stigma is always again sticky. Flowers will be large, colorful, and they will produce nectar as well. Because certain pollinators, such as in Amorphophallus and Yucca case, what happens? The Amorphophallus gives the Yucca a safe place. The flower of Amorphophallus will give the moth Yucca to lay its egg, nurture them, and to hatch their eggs as well. What is Yucca doing in return to Amorphophallus? Transfer of pollen grains. Is this clear for everyone? Yes. Next is the outbreeding devices. What are all the outbreeding devices? So what does this outbreeding devices mean? Outbreeding devices means if a plant is similarly undergoing or daily undergoing or continuously, that's actual term, continuously undergoing self-pollination that can lead to the destruction of the plant or loss of reproductive ability of the plant. So in order to avoid that, plants itself will take out different methods that is known as the outbreeding devices in order to discourage self-pollination and encourage cross-pollination. Clear? Because continuous self-pollination will lead to inbreeding depression, The very first, pollen release and stigma receptivity are not synchronized. That is, pollen will get released at a time, stigma will become receptive at another time. So there won't be any synchrony between them. So as a result, what happened? There will not be any kind of self-pollination happening. Anther and stigma are placed at two different positions. Okay, that is, at any cost, anther will not, that is, pollen grain will never fall on the stigma of the same flower. Inhibiting pollen, this is actually taken by chemical interaction by the female plant itself. They won't allow the pollen grain, even if it is the correct pollen also, they won't allow the pollen to germinate on there. Okay, so inhibiting the pollen pistil interaction, and finally the production of unisexual flower. Okay, they'll start producing only a single type of flower, either male or a female flower. So there won't be transfer or happening of what? Self-pollination. Is this clear? Yes. So what are the methods over here? That is a pollen release and stigma receptivity are not synchronized. Anther and stigma are placed at different position, inhibiting the pollen germination in the pistil production of what? Unisexual flower. What is it? Production of unisexual flower. The pistil has the ability to recognize the correct pollen, that is, you will use a biological term, compatible pollen, to initiate the post-pollination events that leads to fertilization. And finally, what's happening? The pollen grain produces what? The pollen grain produces what? It will produces the pollen tube through which the male gametes will be transferred inside the embryo sac. Clear? So that's all about the pollen pistil interaction as well. Clear? Next methods is the artificial hybridization. I'll tell you how many slides are left out. 5 more slides are left out in this chapter to get it completed. Clear, everyone? Yes. What is artificial hybridization? Artificial hybridization means I have observed a very beautiful red flower over there, and by looking at the another side, I observed a very beautiful pink flower over there. So I have to get an intermediate. I love both the flower. I love both the color of the flower. So I just imagine how beautiful will be the flower that consists of the breed between this red and the pink. That will be tremendous, right? Beautiful one. So I wanted to make it. So I'm doing the artificial hybridization technique. So I'll be collecting the female part or the female gamete from one part, or I'll be selecting the red kind of flower as the parent, and I'll be testing out the pollen grain from the pink onto that. So I'll get the intermediate of both red and pink color flower. So how is it done? Firstly, I'm going to emasculate the flower. That is, from this red flower, if any kind of male gametes or male parts are present, I'm going to remove all the male reproductive structures from the all kinds of male reproductive structures from this red flower, and I'm going to collect the pollen grain, and I'm going to cover the red flower with a plastic cover or a bag, and that process you will call them as the bagging, in order to avoid the contamination of unwanted pollen grains in that desired stigma. Next, I'm going and collecting the desired pollen grain from that pink flower and going to test the same pollen grain from the pink flower into this red flower, and finally I'm going to surprise with the final product, okay, by the artificial hybridization technique. So what are the methods over here? Emasculation, bagging, and desired pollen grains into the nesting of the desired pollen grains into the stigma. Clear? Clear, everyone? Yes. So let's move with the next part. Double fertilization event. What is actually double fertilization event? Double fertilization means we have already told that the male gamete, one male gamete is going to get fused with the egg. And that process of fusion of male gamete with the egg, you will call them as the syngamy, and the other male gamete fuses with the polar nuclei in the center, which you will call them as the triple fusion. So the process of syngamy, fusion of sperm and the egg, plus polar nuclei and the male gametes, which is a triple fusion, since two different fertilization or fusions are happening, you will call the process, the entire process, as double fertilization. That's an easy one, right, everyone? So what is actually the product? What is actually the product of what is actually the product of syngamy over here? That is, a male gamete is going to get it fused with the egg, and what is its product? Zygote, definitely, right? Or the embryo, definitely. Whereas what happened in case of triple fusion, that is a fusion of the other male gamete with the two polar nuclei present in the center, which you will call them as a triple fusion. What's its product? It will result in the formation of a primary endosperm nucleus. What is it? Pen or primary endosperm nucleus. Since two types of fertilizations are happening in case of an embryo sac or inside an embryo sac, you're calling this entire process as double fertilization. Just tell me yes or no on the chat box if you have got those points. Yes. And what is the ploidy of the angiospermic endosperm? The ploidy is 3N. What is it? It is triploid, or it is 3N condition. Suppose if in the exam a question can come, what is the ploidy of the synergid? I'll write the ploidy of everything that have been mentioned over here. Okay, so listen very carefully over here. Synergid. Ploid is haploid N. Pollen grain N. Male gamete N. Egg N. Polar nuclei. Suppose in the option if N only given, then you can write N, or N plus N, or 2N also you can write. Clear? Primary endosperm nucleus or endosperm, it's 3N. Zygote, it is 2N. Clear? Clear everyone? So pollen grain or the male gamete, it is N. The egg, it is N. Zygote, it is 2N. Endosperm, it is 3N. Very important. Apart from all other labelings or all other ploidies, endosperm ploidy is the most important one. Clear? After coming on to the double fertilization, the next topic that you're going to come across is the post-fertilization events. Okay, so we have already done the formation of endosperm, and now we are going to have the endosperm development, embryo development, maturation of the ovules into seeds and ovary into fruit. Endosperm is actually triploid in nature. There are mainly two types of endosperm. That is free nuclear endosperm, which is a common type, and the cellular endosperm. Okay. Yes. So the free nuclear endosperm, which is a common type of endosperm, and the cellular endosperm. This is again important, guys. So please pay attention towards the same. Albuminous seeds and non-albuminous seed. Non-albuminous seed and albuminous seed, or albuminous seed and non-albuminous seed, that you can either call them endospermous seed and non-endospermous seed as well. Clear? Albuminous seed means a portion of the endosperm is remaining inside the mature seed, for example, wheat, maize, and castor. Whereas in case of pea and groundnut, there is no endosperm for the further development. Endosperm is completely developed, or sorry, endosperm is utilized completely by the developing zygote. Clear? Is this process clear for everyone? Yes. Clear, everybody? So, coming on to the embryogeny process, embryo consists of an embryonal axis, a plumule that will develop into future stem, radicle that is developed into a future root. Monocotyledonous seed, the cotyledon of a monocot seed or a maize grain, you will call them as a scutellum. Very important. Maybe you might not understand the question. That is the very important question. The cotyledon of a monocot seed, you will call them as the scutellum. Okay, the part, the undifferentiated sheath that is covering the radical portion, you will call them as what? The coleoptile, and those covering the plumule, you will call them as the coleoptile. Okay, coleorhiza and the coleoptile. Okay, coming on to the last and the final topic of this chapter, seed. Coming on to the seed, you know that what is seed? It is a fertilized ovule. Fertilized and a mature ovule, you will call them as a seed. And a fertilized and a mature ovary, you will call them as a fruit. Okay. So seed mainly consists of a cotyledon. That is, it may be a cotyledon in case of monocot, two cotyledon in case of a dicot as well. An embryonal axis, seed coat will be there. That is, seed coat is actually what the integument will convert into seed coat. Testa, which is the outer layer, and tegment, which is the inner layer. Micropyle will remain as a small opening on the seed coat which facilitates the entry of water and oxygen. Hilum is a small scar on the seed coat, and seeds can be albuminous or non-albuminous. What is perisperm? That's very important. It's a remnant of nucellus that is persistent. That is a remnant of persistent nucellus. Example, black pepper and beet. Okay. Dormancy is actually the term used to indicate the state of inactivity. The wall of the ovary, you will call them as the pericarp. Clear? And when you have already studied about the parthenocarpy as well, but here you are studying about a different concept, that is the apomixis. That is, it's a form of asexual reproduction that is actually mimicking the sexual reproduction. That is formation of seeds without fertilization. You'll have the seed formation. There won't be what? Fertilization. Clear? Sometimes it can be developed by the cells of nucellus surrounding the embryosac can protrude. Example, in citrus and mango, so there can be more number of embryos, so that you will call them as polyembryony in citrus and groundnut. So that's all about this super amazing class. Tomorrow you'll find with the next chapter. Thank you everyone. Take care.